going to talk about the diversity of cells and cell division. The topics we are going to cover today will include comparison of important cell types, cell division and its significance. We will cover the process of one of the cell divisions called mitosis. We will talk about the significance of mitosis, various errors of mitosis, why mitosis is required, and what are the results if there are some errors in the process of mitosis. First of all, we will talk about the comparison between various cell types, important cell types. As we know that animals and plants are two important divisions of the living organisms. Animals, in comparison to plants, are more complex organisms. They have to perform more functions and more complex functions. Plants, in comparison, have simpler structures. This is the reason that the cells of animals and cells of plants, they have certain differences, but of course they do have certain similarities. We talk about the differences and then we'll talk about the similarities. Animal cells. In animal cells, the cell membrane or the plasma membrane is the outermost layer, but in plant cells there is a cell wall that surrounds the cell membrane, which means that plant cells have their outermost layer, the cell wall, but in animal cells the outermost layer is the cell membrane or the plasma membrane. In animal cells there are many vacuoles, food vacuoles, vacuoles for storage or other functions. In plant cells, however, particularly mature plant cells, they do have a single very large vacuole that covers almost all volume of the cell. This large vacuole is generated or developed by merging of smaller vacuoles which were present in the immature cell. The animal cells, they are roughly round in shape, or maybe they have diverse types of shapes. For example, a human body have about 200 types of different cells. Animal cells, they also have various different types. For example, a nerve cell have a particular structure. A muscle cell have another type of structure. The cells of stomach have a different types of structure. But the plant cell, due to presence of cell wall, have a roughly rectangular structure. The animal cells, they have their nucleus present almost inside the center. In the plant cells, in contrast, they have their nucleus, in mature cells particularly, on side of the cell because of presence of a very large vacuole. The nucleus is put aside and this is not present in the center. So this is another difference, and that difference is due to the presence of large vacuole inside the plant. In animal cell, cilia are usually present, but in the plant cells, cilia are rarely present. Animal cells do have centrioles, which we know act in the cell division. Centrioles are not present in most of the plant cells, but in fewer lower plants, centrioles are present. Now we look at the diagram of an animal cell and the plant cell. We can find out so many similarities. As we know that animal and plant cells both are eukaryotes. As the diagram shows, you can see that both have a nucleus in the center, which is covered by nuclear membrane and which have nucleolus inside, chromosomes, the chromatin material inside, and both of these have a cell membrane. Though in plants, this cell membrane is covered by a cell wall. Both of these cells, animal and the plant, have mitochondria, many mitochondria, which we know are the powerhouse of the cell, and they produce energy in the form of ATP for the cells. Both of these have ribosomes, which actually produces the proteins for these cells. Both of these cells have endoplasmic reticulum, which actually modifies the proteins, particularly in the three-dimensional structure of proteins. They play a good role. Both of these cells have Golgi bodies or the Golgi complex, which actually is the post office of the cell, which packages different materials inside the cells, for example, proteins. As we see, both have cytoplasm. Both of the cells have a fluid that in which all the colloidal material, the organelles, the nucleus, and everything is present. Both of these cells have cytoskeleton as well. So the plant and animal cells have so many similarities and some differences based upon the functions they have to carry out. Now we finished talking about the animal and plant cell, and we are going to talk about the difference between prokaryotic and eukaryotic cells and then similarities. Prokaryotic, as the name says, pro, initial, ancient, old type of nuclear material. Eukaryotic, true nucleus. The prokaryotic cells actually do not have a defined nucleus. Though their nuclear material, their chromatin material, is present in almost center of the cell and do not move from its place, but this is not covered by a nuclear membrane. So we cannot call it a nucleus. We say that prokaryotic cells do not have a defined nucleus. But the eukaryotic cells, they do have a defined nucleus covered by a membrane that is called the nuclear membrane, which separates the chromatin material from the cytoplasm. The second important difference is that prokaryotic cells do not have membrane-bound organelles like endoplasmic reticulum, but the eukaryotic cells, they do have membrane-bound organelles. In prokaryotic cells, they are usually small in size in comparison to the eukaryotic cells. Their size usually ranges from 1 to 2 micrometers. But the size of most of the eukaryotic cell on average is fairly large in comparison to the prokaryotic cells. They are about 20 to 30 micron in size, but their sizes do vary. But on average, eukaryotic cells are larger than the prokaryotic cells. For example, we know that bacteria, the commonmost bacteria, their size is about 1 to 2 microns, that is 1 micrometer wide and about 2 micrometer long. But the animal cells on average, for example, their diameters is roughly 20 micrometers. The next difference is this, that the prokaryotic cells have a cell wall and eukaryotic cells, not all of them, animal cells don't have a cell wall, but the plant cells and the fungal cells, they do have cell walls. But there is a difference between the prokaryotic cell wall and the eukaryotic cell wall. The prokaryotic cell wall consists of a polymer of amino acids and sugars, which is called peptidoglycan. But the cell walls of the eukaryotic cells, the plant cell, consist of large fibers of cellulose molecules, and the fungal cells, they do have a cell wall of chitin. So cell wall is present in both, but it consists of different components. Its chemical composition is different from each other. Now we look at the a diagram for comparison of a bacterial cell with an animal cell. In the diagram, you can see there is an animal cell on the left and there is a bacterial cell on the right. In the animal cell, you see that there is a clear-cut nucleus which have a membrane surrounding it, which is separating the materials of the animal cell of the nucleus, the chromatin material, with the cytoplasm. In the bacterial cell, you can see that there is a region in which the chromatin material is present, but there is no membrane. This region is called nucleoid region. The chromatin material is present in this region. Though bacteria, prokaryotic cells, do have another type of DNA which is present in the cytoplasm, and this is in circular form, and this is called plasmid. Plasmids are very important parts of DNA because these are used in biotechnology. These are used when we want to insert, for example, a human gene into a bacteria, and then we want its mass production on large scale. We want to make multiple copies, because bacteria, when they divide, plasmids also divide, and we can get many plasmids and many copies of that human gene or an animal or plant gene that we want. We can see that both have a cell membrane. So here they are alike. They have a cell membrane. They have a nuclear material, chromatin material, and both have ribosomes, which are alike. Now we finished talking about the prokaryotic and eukaryotic cells, their comparison. Now we go ahead towards the cell division. Cells need to grow. Organisms, they need to grow. They need to increase in their size sometimes. Sometimes they are wounded and they need healing. They have to increase in number. We call this cell division. There are two types of cells if we are considering the division. So we'll talk about, there are some cells called somatic cells. There are some other cells called germline cells. Somatic cells are most of the cells of the body other than the gametes. The germline cells are the cells which are called gametes, which are present in the reproductive organs, testis and ovary of the males and females, and these are the cells which are responsible for making the next generation. We are talking about the division in somatic cells first. The somatic cells need to divide for growth, for wound healing, for regeneration or other mechanisms. These cells divide by a process called mitosis, while the germline cells, they divide by another process called meiosis. In today's lecture, we are going to talk about mitosis. In the diagram above, you can see a dividing cell. This is actually, I must not say a diagram, this is a picture that is taken through microscope, and these are the onion cells, onions epidermal cells. You can see when you cut down an onion, you can see a small membrane. These are the cells of that membrane which are dividing. Before talking about division of the cell division, we have to talk about that what is the whole process of dividing. When cells are produced, after that, for some time, they normally perform their function. When they need to divide, they have to enter a cyclic process called cell cycle. What is cell cycle? Cell cycle is the sequence of events by which a cell divides and then go back to the non-dividing state. The cell cycle consists of two major phases. One is called an interphase, which is actually a very long phase, and it consists of three subphases called G1 or gap 1, S phase or the synthesis phase, and G2 or the gap 2 phase. After the interphase, next phase is the M phase or mitosis phase or the division phase. Interphase is actually a preparatory phase for the M phase because cell need to prepare itself for division before it enters into the division phase. The G0 or the gap 0 is also called a resting stage. This is the stage when cell do not divide. Here you can see a diagram which shows the interphase and the mitotic phase, the M phase. As you can see, that interphase makes about 80 to 90% of the cell cycle, and the remaining small phase is called the mitotic phase, M phase. Interphase, as you can see, is again divided into these three phases as I have described: the G1 or gap 1, the S or the DNA synthesis phase, the G2 phase, then comes the M phase. After M phase, just at the start of G1, cell can enter into the G0 phase or the resting phase and stop dividing. If cell enters into the interphase, then it means it is going to divide. If it is not going to divide, it is genetically programmed to not to do so, then it's staying in the G0 phase. For example, our nerve cells, they do not divide. They stay throughout their lifetime, throughout our lifetimes, in the G0 phase. This is the reason that if there is a injury to the nerve cells, they are never replaced because these cells do not divide for wound healing or for replacing themselves. Now we look at the details of these phases. The gap 1 or G1. In this phase, this is the first phase of the first part of the interphase. In G1, cells increase in size. And as we know that after G1 phase, the next phase which is coming is the S or the synthesis phase. Synthesis is the DNA synthesis phase in which DNA have to be synthesized. In the gap 1 or G1 phase, cell prepare itself for the DNA synthesis. We call it replication or duplication. the DNA of the cell have to be duplicated so that the cell produces those materials which are required to make the new DNA, to duplicate or replicate the DNA. There is a G1 checkpoint. There is a control mechanism. This control mechanism checks, or G1 checkpoint checks, that whether all the materials which are required for entering into the S phase, the synthesis phase, are they ready or they are not. If all these materials are ready, then cell enters into the S phase. If they are not, cell have to complete making of those elements. For example, if an enzyme required for DNA synthesis is not formed or is formed in lesser quantity, then G1 checkpoint stops cell from entering into the S phase, and it will make the enzyme and then enters into the S phase. The S phase is the phase in which the DNA is synthesized and the cell actually duplicates its DNA. As we know, that DNA is present in the form of chromosomes, and chromosomes are present in pairs. For example, human beings have 46 chromosomes. These 46 chromosomes are present in 23 pairs. All of these pairs, because we know that mitosis is a division which will make two daughter cells, and these daughter cells should have an equal number of chromosomes. This is the reason that in the S phase, before division, cell have to duplicate all of these chromosomes so that both the daughter cells should have an equal number of those chromosomes. So in synthesis phase, DNA is synthesized and all of the chromosomes are duplicated. They are actually double to their initial number. There is an S check. When the DNA is duplicated, this checkpoint or the control mechanism checks that whether the DNA is duplicated and is it correctly duplicated. If this is not, cell have to redo these things. If cell is not able to do so, it is stopped. When it completes its things which are required for the duplication, and duplication is complete and correct, then cells enter into the gap 2 or G2 phase. Now comes the G2, the gap 2. In this phase, cell again continue increasing its size, that is, it grows. And as we know that after gap 2 phase, the phase which is coming is the mitosis, the division phase. So in gap 2 phase, cell collects all those materials which are required for division. The most important element, the most important thing which is made or produced during this phase are the microtubules, because microtubules are required to move chromosomes, to attach with the chromosomes and then move them correctly towards the poles of the cell. There is a G2 checkpoint which checks whether all the materials required for the M phase complete or not. If they are complete, checkpoint allows the cell to go ahead towards the mitosis or the division, or if they are not complete, then the control mechanism stop the cell over there and cell have to remake all of its materials. When it completes, the cell enters into the M or mitosis phase. Now mitosis, M phase, the division. In mitosis, basically mitosis, in the mitosis, cell have to be duplicated. Cell have to divide into two daughter cells. At this division, cell growth is totally stopped. As we know that in G1 and G2, cell was growing, but in mitosis phase, cell stops its growth, because for growth, cell needs to apply energy, and for division, the cell division is a process which requires a lot of energy. So cell put all of its energy for division only, and it do not grow during this phase. There is a check when the cell is dividing. There is a checkpoint almost at the end of the mitosis, which checks that whether the mitosis is correctly carried out or not. If this is not, cell is stopped from entering next phase, from dividing, If it is okay, cell go towards dividing into two. Now we look at the diagram. This diagram in front of you can show quickly the whole process of mitosis or the division. As you can see in the first diagram A, the cell is present in the interface. The nuclear membrane is complete. And now this is the diagram showing the mitosis in animal cell. You can see a nucleus in between. You can see centrosomes, a pair of centrioles present on one side of the nucleus. The diagram B, next one, shows the early prophase. In this diagram, you can see that the chromosomes, they start appearing. They become condensed, and we can observe them under a microscope. The spindle fibers are forming. After that, cells enter into the next we can call it the late prophase. In late prophase, the spindle fibers are formed, and the chromosomes start attaching themselves to the spindle fibers. After this, next stage comes the metaphase. In metaphase, chromosomes arrange themselves on the spindle fibers. They are attached to spindle fiber almost in the center. In the next phase, anaphase, the chromosomes starts moving towards the poles of the cell, and they are, as you can observe, the chromosomes, they are divided from the center, and equal number of chromosomes are going on both sides. In the telophase, you can see that the chromosomes reached that center. They started thinning again, and the nuclear membrane starts forming. In the next phase, you can see that the cell is divided into two daughter cells. Now we look at these processes, all of these stages in detail. Mitosis results in two cells which are alike, just like each other, by almost every aspect. Mitosis is also called, it's a karyokinesis. You know that karyo is the word used for the nucleus. This is for division of the nucleus. When the mitosis of the karyokinesis completes, then comes the next phase called cytokinesis, when cell itself is divided into two. The karyokinesis consists of 4 phases: prophase, metaphase, anaphase, and telophase. When these 4 phases complete, prophase, metaphase, anaphase, and telophase, then the nucleus is completely divided, and then the cell divides itself into 2 daughter cells. Now, cytokinesis is different in animal and plant cells. We will talk about it later. We are talking about the actual mitosis, the karyokinesis. Prophase, we are talking about the first phase, the prophase. In the interphase, the chromosomes are not visible. We can see only the nucleus because the chromosomes are very thin at that stage. In prophase, chromosomes start condensing. They are, due to the condensation, they become appearing under the microscope. Prophase. First of all, we are talking about prophase. In the interphase, chromosomes are very thin and they are not visible under the microscope. But as the prophase starts, the chromosomes start condensing, start thickening. Due to this condensation, they become visible under the microscope. As we know, that cell has passed through a process of DNA synthesis already. So each chromosome at this phase is duplicated, is double, and have 2 parts which are alike called the sister chromatids. If you look at the diagram above, you can see that one chromosome is duplicated and it is looking like there are 2. These are actually the replicated or the duplicated chromosome. Now each chromosome have a central point where we call both of these parts the sister chromatids. They have a central point, the centrum here, where both of these sister chromatids are joined together. Then this part is also occupied by a protein called kinetochore. Kinetochore is actually attach these sister chromatids to each other, and later on in the later prophase, when the spindle fibers are formed, kinetochore attaches these sister chromatids to the spindle fibers. In next part, in the prophase, the chromosomes, they are condensed. They consist of sister chromatids attached to each other. The the centrioles, they start moving towards the poles of the cell, and they form the spindle fibers, long fibers, which are present for the attachment of chromosomes and their movement. At the end of the prophase, the chromosomes attach themselves with the help of their kinetochores to the spindle fibers. At the end, when this is complete, the nuclear membrane start dissolving, and now the nuclear membrane is not present, so the chromatin material is exposed in the nucleoplasm only, and they are not surrounded by a membrane. The nuclear membrane disappears. In plant cells, the centrioles are not present, and the spindle fibers, they are formed with the help of the components of spindle fibers, the microtubules present inside the cytoplasm, which are produced by the endoplasmic reticulum and the Golgi bodies. Then comes the next phase called metaphase. In metaphase, chromosomes arrange themselves on the equator. As we know that in the prophase, chromosomes, they were attached to the spindle fibers with the help of their kinetochores. Now they arrange themselves in almost center of the cell. We call it equator. So in metaphase, cells arrange themselves in the center. After metaphase, the next stage is called anaphase. In anaphase, the spindle fibers start pulling chromosomes towards the poles. When they apply the pulling forces on the chromosomes, the sister chromatids divide from their kinetochores and they are separated. They divide actually from the centromere from which they were attached to each other, and the sister chromatids, they are separated, and out of each chromosome, one sister chromatid move towards one pole and the other sister chromatid move towards the other pole. This stage is called anaphase. Then these chromosomes keep moving towards the poles in the anaphase, and this stage is important and critical because equal number of chromosomes actually in this at this time, the sister chromatids should reach each pole to retain the natural chromosome number. If some sister chromatids, they do not divide, then the result will be faulty cells, and they may die. They cannot survive. So the anaphase is the phase in which the spindle fibers start pulling the chromosomes towards the poles. Chromosomes are divided at their centromere, and each sister chromatid move towards one pole. Then comes the next phase called telophase. In telophase, the sister chromatids reaches at the poles. At this stage, they start decondensing, and they start, we can say, thinning again, coming back to their normal position in which they were present before the prophase. At this stage, we can say the nuclear material, the chromatin material, the chromosomes, they start decondensing. Also, at the end of this stage, the nuclear membrane start reforming. When this stage reaches the mitosis, or we can say the nuclear division, the karyokinesis is complete, and then the cell enters into the next phase called cytokinesis. That is division of the whole cell. Now, division of the whole cell is different in the animal and the plant cells. We have a look on certain pictures taken from onion cells in their prophase, in their metaphase, anaphase, and telophase. Let's have a look on these pictures first, and then we will talk about the cytokinesis. Here in the diagram, you can see in the circle on the top, prophase. This is the real picture taken from the microscope. You can see that chromatin material is condensing. On the right down, you can see a metaphase. The chromosomes are arranged on the equator. In the next picture, you can see a beautifully placed anaphase in which the chromosomes are moving towards the poles, and they just look like two umbrellas facing each other. In this diagram, again in the circle, you can see a telophase in which the chromosomes have reached their destination to the poles, and they start decondensing, but yet cell is not divided. After this karyokinesis, nuclear division is complete. Now cell have to divide in two cells. Now the cytokinesis or division of the cell is different in the animal and the plant cell. We talk about the plant cell first. In the plant cells, as you can see in the diagram above, in the center of the cell, a plate or a line is formed. This plate is formed of some materials which are produced by the Golgi bodies of the cell. These materials start collecting inside the center when the nuclear division is complete, and then this cell plate start moving towards, or we can say start growing towards the sides of the cells, and ultimately it joins with the cell membrane and divides the cell into two. Now this cell plate have components of cell membrane in the cell wall, so that it can make the cell membrane to separate the cells and then the cell wall as well. After that, the cell divides into two different cells. Cytokinesis in animal cell is different. As you can see in a picture above, when the nuclear division is complete in the animal cell, a cleavage furrow is formed. We can say an invagination is formed in the center of the cell, and this invagination continue to grow towards inside. As we know that in plants, the cell plate was formed in the center and then it moves towards the external sides. But in the animal cells, a furrow or an invagination is formed in the center from the exterior, and then it is moving towards or growing towards the interior of the cell, and then it divides the cell into two. This process of cytokinesis in the animal cells is supported by the cytoskeletal elements. The actin filaments and microtubules and other cytoskeletal elements help in this cleavage furrow formation. They actually contract and stretch the cell membrane, the plasma membrane, towards inside, and ultimately this invagination from all the sides join each other and divide the cell into two daughter cells. Now the division of cell is complete, and the both cells are now called daughter cells. These cells have equal number of chromosomes and almost equal quantity of the cytoplasm and organelles and all the other things. So this is the process of cell division in the plant and animal cells, the somatic cells. Now we will talk about the importance of cell division. Why cell division occur? There are three major objectives: development and growth, regeneration, and asexual reproduction. Cells need to divide. There are few parts of our bodies or animals' bodies which do not grow. They do not require a cell division. As we previously talked about, the neurons and nerve cells, they do not divide. They remain in G0 phase throughout their lives. So they do not divide. They do not need division. But many other cells of the body, they require a continuous growth. For example, our skin cells, they need continuous growth. Our skin cells are continuously falling down and new cells are forming. Cells in our stomach, they are also very important cells, and they are exposed to different pHs due to entrance of the food, its digestion, and other processes. So the cells of stomach need to be replaced again and again. In all of these tissues, like the stomach, in our mouth, our cheek cells, on our skin, and many other cell types, they need to grow. And for growth, they need to divide. And this is very important, that they divide in same type of cells with same number of chromosomes. So mitosis is a very, very important process which is required for the tissue and the organ growth. For the tissue growth, for the growth of the organism as a whole, and so on. There is another requirement. If sometimes organism's body is wounded, it is injured. For example, we hit our hand on some hard surface and our finger is injured. An animal has injured. When it is injured, its skin cells are cut off, its muscles, they are broken down or something, and they have to be replaced. for wound healing, we also need cell division. Just like that, many times the plants, they are also wounded, eaten up by, for example, an animal, cut down by a kid or otherwise. They also need to replace their parts. So mitosis is very important in the growth of the tissues and in the wound healing. There is another very important process in which mitosis helps. Few organisms can replace or remake their lost parts. For example, a starfish, which have usually 5 arms. If its one arm is broken or cut off, or we can say amputated by a predator, it can remake its whole arm by the process of, we call, regeneration. It can regenerate its whole arm. It is able to regenerate the arm with the help of the cell division or the mitosis, because we know that mitosis is the cell division in the somatic cells, the cells other than the germline cells. In some other organisms, the regeneration also takes place. For example, the lizards, they also can replace their lost tail. Many times what happens that a lizard is escaping from its predator. It is successful to escape, but the predator somehow breaks down its tail. Lizards also replace their tail with the help of mitosis, the cell division. So it means that organisms can divide their cells to produce new cells for the tissue growth, for the wound healing, and for the regeneration of their lost parts. Cells also use mitosis. Few organisms also use mitosis for the asexual reproduction. There are few unicellular organisms which asexually reproduce by the help of this simple cell division, the mitosis. So mitosis or cell division is a very important process. Now we see that what happens if there are errors in mitosis. There may be different types of errors in the mitosis of cell division, like particularly, mostly, what could happen that when the chromosomes divide at the centromere during the anaphase, when the chromosomes, of course, we know that each chromosome at that time consists of sister chromatids, and for going towards the poles, it have to be broken apart, or it have to be cut off from the centromere into 2 sister chromatids, and each sister chromatid have to go towards one pole. If sometimes or somehow by a mistake they are not cut off, they are not broken at centromere, and the whole duplicated chromosome goes towards one side, then in one cell there will be more chromosome, in one daughter cell there will be more chromosome, and in the other daughter cell there will be less. Most probably these cells will not be able to survive. Now this process of cell division is highly regulated, as we have seen in the process of mitosis and in the process of cell cycle particularly, that there are different checkpoints, controlling mechanisms which always check that whether the cell has completely or correctly completed the process as required. For example, during the G1 checkpoint, which checks that whether all the materials required for DNA synthesis are complete, or before the M phase or the division phase, in the G2 phase, are all the materials which are required for the mitosis, the karyokinesis, are they complete? But sometimes what happens, and in the M checkpoint, the mitosis checkpoint itself, which checks that whether the nuclear division is correctly completed. But sometimes what happens that this regulation fails. And if somehow at some place this regulation is not correct, then the result will maybe uncontrolled growth of cells. The worst sequence is the uncontrolled growth of cells. If the cell cannot stop dividing after M phase, it do not enter into a proper G0 phase and rest for some time and then go to the interphase as normal, but it grows randomly. Sometimes it happens due to mutations. There are certain types of mutations due to which mitosis, the process of mitosis continues uncontrolled, and the checkpoints, they are not properly checking their places where they have to check that whether everything is correct. The result is this is the uncontrolled growth of cells, not in the form of normal layers of cell, but in the form of heaps. हम ऐसा भी कह सकते हैं कि जिस तरह से सेल्स जो हैं वो एक ढेर की शक्ल में बन जाते हैं, बजाय इसके कि जैसे वो नॉर्मल लाइंस बनाते हैं उस तरह बनाएं. The result of this is tumors. We call these heaps of cells tumors. If these tumors are just present, but they are not like growing further, or they are start damaging other tissues. And second thing, they do not break off from their places. जो cells के ढेर हैं वो उसी तरह रहें और दूसरे इर्द गिर्द के जो टिश्यूज हैं उनको ख़राब ना करें और अपनी जगह से टूट कर दूसरे टिश्यूज में या ब्लड में या और जगहों पर ना जाएं, तो हम इनको कहते हैं बिनाइन ट्यूमर्स. दीज ट्यूमर्स आर कॉल्ड बिनाइन ट्यूमर्स. बिनाइन का मतलब है कि जो नुकसान नहीं पहुंचाते, हार्मलेस ट्यूमर्स. बट समटाइम्स व्हाट हैपन दैट दीज ट्यूमर्स they become, we call malignant, they become cancerous. They start growing very fast, their size increases a lot, and the cells from these tumors, they break off and go to the blood circulation, and through blood circulation they reaches other tissues and makes tumors there as well. So when these cells which are broken off from the original tumor, they go to different tissues and they can make secondary tumors at these places. We call this state or situation metastasis, which is the spread of the cancerous cells or the cancer, which is the very dangerous stage at which treatment of this cancer become very, very difficult. So uncontrolled growth of cell may result in very serious consequences. Now we finished talking about different comparison of different types of cells, and then we talked about the cell cycle, which is the sequence of events in which a cell divides, and we talked about the division in the somatic cells, the mitosis. And then we looked at the significance of mitosis, why it is required, and what could be the result of various errors in the mitosis. I hope you understand what we talked about today. We will meet in the next lecture. Thank you.